All right, so welcome. So today, I'm going to give a talk about some of the research that I've been involved with, and we're going to talk a little bit about machine learning, but then how that connects to other things and how you might, kind of answer questions and things like that. So I'm going to go over some projects that I've worked on in my research. Today, I'm going to talk about machine learning and how use this in computational biology or two different model organisms. So the way this, tends to work, I work in computational biology, specifically developmental biology, but also other areas of biology. And in biology, we usually have model organisms that we use to understand biological systems. So, you could look in humans, but sometimes that's too complicated of a system to really get a good handle on for a number of reasons. So a lot of people are interested in humans, but a lot of people are interested in other organisms. And again, sometimes those organisms are hard to examine outside the lab for specific reasons. And so what we want is an organism that we can look at under the microscope. We can culture in the lab. We can put in a little dish or in a small tank of water, and they can reproduce, and we can have a captive colony so we can do things kind of under, strong control. So we don't want to have, like, we can't really image things like in the wild. And so we want to have model organisms that we can do things with and that we have a lot of prior data. So I'm going to talk about C. elegans, which is this nematode top right. And that is a small worm, a very small worm, only a couple millimeters in length. I'm also going to talk about diatoms, especially diatom basilaria, which is, these are both computational models. We'll see real organisms in the slides. And again, this is a small organism that lives in water, in the water column, and they propel themselves in a unique way. So one of the reasons why we're focusing on these two organisms is because we're able to culture both of them. We have very detailed data that we can use. We have behavioral data, we have genomic data, we have other types of data that we use in conjunction. And so we can bring those data sets together to understand, to really answer questions in biology. So we're looking in the nematode C. elegans, specifically at the developmental biology, and in basilaria, which is this diatom, at the biophysics of its movement. So we can answer specific questions about these systems. We have the data available. And so that makes them amenable to doing these machine learning types of analyses. So this is a diatom. Diatoms are actually quite diverse. So diatoms are these single-cell organisms. They're eukaryotes, so they're not like bacteria, which are prokaryotes. They're eukaryotes, meaning they have these cells of organelles in them. You know your biology, we're also eukaryotes. And so these are just single-cell versions of, or they live in small colonies where they have these varying shapes because they have these walls of silicon, which is this hard material. So they form these very intricate shapes. So we know the anatomy of diatoms pretty well. You can actually image them. These different see these variations on the lower right. These are at the size scale of 10 to 80 microns. So it's very small, and you need powerful microscopes to look at them and really examine them in detail. But there are all sorts of different shapes, rod shapes, plate shapes, and various 3D shapes. Now, that being the case, there are a lot of different modes of locomotion, and we'd like to understand that locomotion to be able to predict it, and then be able to predict the mechanisms of that locomotion. So that's and then of course, being a very small organism, we can really kind of get down into the details of what makes something, move. And so these are some videos. This is, if we were to go and look for diatoms in the wild, we might look at a river or some other environment, sort of at the top of the river or on the edges, something like that. and you could like scoop up some water, isolate the, put them under the microscope. And this is what they look like. These are these Basilaria diatoms. So they're these rod-shaped diatoms. So each of these little things here is a cell, right? And this is just them moving around in the water. We'll take model water from your microscope slide and then do this movie where we have, they're kind of stacked up together like this, these little rods, and they slide like this. So they have like, they slide out so they can form a structure like this. And then they kind of form this accordion that kind of moves back and forth. So it's a very unique mode of movement. And we wanted to understand how to model these colonies of cells and then how to model the movement. That's in the wild. So this is kind of just looking at a bunch of them under a microscope. This is a single colony. So this is in more detail. This is actually what we used for some of the stuff I'm going to show you here. This type of thing where it's fixed. And, you know, you have like, you can see the movement in a very unobstructed manner. So everything's in the same focal plane, meaning you see everything without any sort of blur. It's moving back at depth. So it's not doing that, it's fixed. And it's moving, sliding back and forth. You can see a couple things. We can see that it doesn't go all, it doesn't separate all the way out between the cells. There's something holding them together. And that they're maybe, you know, 4 or 5 in a stack, and that they move kind of like an accordion. You know, the physics are very distinct. So, you know, what if we could take these cells, segment them out of this image, and then once we have that, then we can look at the relationships and model, predict what those will look like. So remember we talked about segmentation, machine, where we take the features out of an image. We have this background that's gray, we have that image, and we want to segment out the thing of interest in the image, make the background. We want to do a good enough job so that we have a highly precise feature and that those features are identifiable by, say, an algorithm. And so, again, we have this accordion-like movement. And in physics, they talk about this as an oscillator. So if you go to the physics literature, a lot of times they'll model this sort of thing as an oscillator, meaning it's kind of like goes around, it has this period, goes around, it has a specific beat to it. So like oftentimes they'll describe rhythm as an oscillator. Or when I'm walking like this back and forth, that's an oscillator. The legs are oscillating, or something like that. So we can describe this as an oscillator. We can also kind of think about the physics of movement. So this is stationary, this is the position of something. And if it's sliding around like this, it's actually has a movement vector that shows velocity and acceleration. So we can extract those parameters as well. Okay, so we can model this system as a series of coupled oscillators. So we have these coupled oscillators that are moving against each other. You know, these cells, you can see kind of the variation, and we don't. I showed you a very isolated example, just one example, but you can see in these colonies that are very complex. You have a lot of them stacked together. You have some of them kind of twisting around. And so the movement's a little bit harder maybe to predict than you would think just by looking at that last video. And that you have maybe you have a basic mode of movement, but it results in a wide variety of things going on. And so sometimes the stimuli that they get, sometimes they detect light, or they detect salinity in the water, whatever, and they react. So there's this aspect of their movement being mediated by where they are in the world. So this is what we call electron microscopy. So this is where you have a very high resolution microscope. And like the images I showed you were kind of hard to see the detail of the cells, are just kind of cells stacked up. This is really high density stuff here. So this is like, these images show these perforations along the edge here, these pores. And you can see like this image looks kind of like the others, but these are very high density. This shows a great amount of detail. So we can use these types of data as well to inform our model. So we could have, you know, not only can we segment out these cells because there are contours along the edges, and we can just simply make some sort of mask, or we can use the features of the image to separate out the features from the background. So this image, for example, our background is gray and it's kind of speckled. And we can say, okay, this background has a certain set of color values we can ignore. So we can take those away. And then we have these cells here. So the cells are not they're not all one feature, they're individual cells. So we can say, well, let's find a contour between these cells, maybe a color value that separates them. So if we do that, then we end up with all these different pieces. And then we can treat each of those as a feature, and we can just sum them up and look at the variation. We have higher resolution images. We can take this image. We can say, this is a cell, but then we have additional data to train the model. So we can get these higher resolution images and say, this is the detail of the cell. What does this look like? And then, so when you're sort of training an algorithm to learn patterns, you want it to learn not just single patterns, but you want it to learn a multitude of patterns that are relevant. So this is a project I did with some students. This is a deep learning analysis in bacillarian colonies. So they took some of these video images and they tried to sort of develop a technique for extracting not only the features, but the physical parameters of the model, and build a model of these organisms. So this is it's kind of blurry, but basically, first thing that you do when you want to deal with image processing is you need to sort of figure out where the features of interest are. And so sometimes that involves, like I said, separating out the background from the foreground, finding contours in the image, but sometimes that just involves you making a mask of something that looks like the feature that you want, and then placing that over the image. So this was where they created a mask like this. It kind of looks like a cucumber, and then you would just apply this mask across the cells. And say anything that looks like this is a feature, and it can't overlap with another feature. And if you do that, you can basically get these shapes. And it might cut off some parts of the cell, but it captures it as that feature. So that's one, there are different ways you can just extract features from images. And so this is where they basically took all these movies, extracted these cells as features, and then they furthermore went beyond the sort of mask to define 3 landmarks on the mask. So, mask can be this elongated shape, but it also has an anchor point in the middle and then 2 endpoints. So you have to match it up, and then you get your feature, but the feature also has 2 endpoints and an anchor in the middle. So now we can, this is what we call a bounding box method, where we say there's some shape in this image, and these are our boundaries. This is how we define the boundaries of the space. And we just place that down and say, where is the centroid of that bounding box? And that's our feature. And then we do that across the image. So they also wanted to derive positional coordinates. So they also not only wanted to know where these bounding boxes were and identify the cells correctly, but they wanted to figure out with what relationship are these different cells. So in order to track the movement sliding against one another, you kind of have to know your starting point. So you could, create this image, you have this image, and then you have this feature, then you have this other feature that's not quite aligned with it, it's off center. What is the relationship between this feature and this feature and this feature and this feature? And so you can actually, if you have, a movie, you have a bunch of images, but those images are dependent on time. So that means that you have a record of these features, not just as they are independently in the image, but that they have a relationship over time. So to model the sliding motion, you get this feature here, and then this feature in the next image in sequence moves even farther away from this feature, and then it starts to move back against this feature. So you capture that movement as a series of features. You're not only capturing the features independently, but you're capturing that movement, that relationship. So that means you can predict the movement, not just the cells. You can say, this is a cell and this is not a cell. That's fine. But you can also say, this is how it moves. And you can actually use that as a feature as well. So this was all done with Deep Lab version 3. So this is something you can implement in a Python package. And then we use some other types of software as well for this. And so this is the result. Again, the bounding box, you have a bounding box, and it cuts off certain parts of the cells. But for the purposes of modeling, we don't care about the sort of, the absolute shape of the cells. We care about capturing that cell in relation to other cells. So we can ignore certain aspects of that cell and just have that bounding box as the relevant feature. So this is actually a graph. So this is abstracting it away from the image and abstracting it directly to two-dimensional space. So this is the image that we're analyzing. It's these, this colony. It's pretty messy. It's embedded in the background. But now we've taken those cells. We used the bounding box method. We found the centroid value in XY coordinates. And now that we've done that, we have our centroids and we can plot those out. So this is what our centroids look like with that column. So now we've abstracted things from that image to this bivariate space. You can see that it has the sort of same shape, I guess, but we're just looking at the centroids of each feature. Okay, and we can do this again for this one. This is a little bit, I guess that's a little harder to recognize the basics. I think this is shifted somehow, but basically it has the same pattern. And then this one is for this space, and the axes aren't necessarily at the same, at the same intervals here, so it's a little bit wider than this. Now, of course, the idea is that these points are going to move over time. So this is just a static representation of this bivariate space. If we want to look at the movement, we need to understand the time. There needs to be a third dimension of time. And these points are going to change with time. They're going to shift around on that XY axis, on the XY axes. And then this is just another image, another example. Okay, so then this brings in another person who worked on this. He's a physicist, and he's in Germany, and he's culturing these diatoms. He's getting them out of the river and like putting them under a microscope and getting little cultures of them going. That means he can grow them in his home in water. You don't have to keep going out and getting them out of the wild, but you can also then just kind of take them out of the microscope when you want. And you can do a lot of images out of this. And so what he was doing here is he's doing a similar thing, but using more traditional measurements. So you can use this kind of data to verify what you're doing in machine learning. So in this case, you're doing traditional measurements of just looking at this image, having a scale bar. So all microscopy images have a scale bar on them. The magnification, kind of the size of the slide. So you basically know these are small distances. And if you have the scale bar, then you can measure distances between things in very small units. So this is one of these diatom colonies that are kind of spread out. Notice they don't separate completely, they stick together sort of a little bit at the ends. And then, the idea is we want to measure different landmarks and verify what we're getting with machine learning. So we have this diatom here, which is a cell. We want to measure from the end of the cell. Remember, we have those landmarks to this point where they're sort of their maximum extent, where they're not okay. So it's stretched out a little bit. And that turns out that that's 57.5 microns, which is here. But then for diatom 2, there's a little bit of variation. It's 59.14 microns. So it's a little bit different. And you should expect a kind of variation. And then that's something that we need to keep in mind when we're building our model, is that you don't always have the same distance. And so you have this sort of, and of course, microns It's a very small measurement. And this is not something that you can measure kind of with a tape measure. And so this is where we can then map these distances to the features in our machine learning data set. We can also build interesting sorts of graphs with this. So this is a phase, what they call phase portrait. So in a lot of dynamical systems, we can build phase portraits of different types of phenomena that are these time-dependent processes. So in this case, what we have is we're plotting position versus velocity. And so in this case, we have two variables that kind of co-vary together, meaning that they're not independent. They're not independently identically distributed. They're linked together in time. And so what happens is as position goes from negative 50 to positive 50, the velocity decreases and then increases. Then as you go from 50 back to negative 50, because remember we're oscillating back and forth, the position increases and then decreases. So you end up with this circular kind of pattern. Notice how much noise there is. It's wavy like this. So we can see that in the pattern or in the space portrait. And this is, some indicator of how noisy the data is. Now we talked about noise before as maybe being undesirable for machine learning. So the question is, in biology, we have a lot of noise. That's just a fact of life. Sometimes noise is good because it's actually active in different processes. In this case, we just expect noise because we have, variations in the size of the cell, slight variations in how they move back and forth and so forth. And so we should expect these little squiggles as noise. The question is, should we normalize this for further analysis? Should we smooth out the data, use some sort of basis function or something like that? Or should we just leave it like this? And then, that's really the question. And a lot of biological machine learning, that's, one of the problems you have is you have this inherently noisy data. Sometimes it's noise, where it's just not as clean data as you'd like. Sometimes it's just because the process is just noisy with respect to sort of this normal distribution regime that we love in machine learning. So that's a problem that we often have. But it also establishes that there is indeed a rhythm here. Now we might use other techniques to analyze this. So we might not use machine learning to analyze this, or at least we might not use, say, like the models that we've talked about already. We might use some sort of that we talked about with respect to the different frequency analyses that we were talking about. So we might use a frequency analysis for this. We look at not sort of the absolute numbers or some sort of categorization or some sort of regression, but some sort of what is the frequency of movement? What is the frequency of these little squiggles, things like that. And that might tell us more. Okay, so this is kind of an interpretation of this phase portrait. So this might be, representative of the end of an oscillation period, the signature of jerkiness, meaning higher order movement or some other type of thing. Now, this is the thing about data too, is sometimes you make interpretations, and this is true, no matter what your analysis. So you look at something interesting, in a graph, you look at a high value, or you look at something that deviates from the norm. You say, that's something interesting, which is fine because that's what you do a lot of times in science and in data analysis. But you have to kind of have a basis for your interpretation. And this is where this expertise goes. We're talking the first lecture about expertise. Now we need expertise. We can't just take data and analyze it and say something about it. We need to know kind of what the area is, and maybe we need to do a little bit of reading about what something is. Maybe there's some reason, but in this case, we don't really know. We have to kind of fly without any sort of guidance because we just simply don't know. So, but this also leads you to sort of next steps, right? So you do an analysis and you say, okay, now I see something interesting here, but I can't say anything about it. I can't make an interpretation about it now. But if I set up another study or ask more refined questions, maybe just about this, or maybe about something else, I can maybe get the answer. So for your projects, you're just asking that first question or first set of questions. But ultimately, what you're going to be doing in machine learning is you'll take a data set, you'll explore it, you may answer some questions, use some models, and then you'll find things, you'll maybe have more questions than answers at the end. And when you have more questions and answers, then you need to keep doing studies and you need to keep refining the answers to those questions. Okay, and then this is where we also have these oscillations and they look like sine waves. So we know that we can use kind of a frequency analysis, kind of a Fourier analysis, where you use sine waves as the basis function. Analyzes that. So it's like there's a clue in what kind of method we can use from the shape of the data, right? We know that like it's amenable, isn't really amenable to regression or class. We could classify it, I guess, but we get more out of it with a Fourier analysis. Okay, so, and this is just kind of looking at the difference between the model, which is a sine wave, and then the actual diatom movement, which is blue. What's interesting about this, like this is like kind of its normal mode of movement. It kind of fits sine waves okay. You could probably build a pretty good predictive model like that using kind of a analysis of these movement of movement. You can find the period of that. This is where the diatom actually stop. So they move around for a while and then they stop. And they just stop in a certain position. And this stopping actually is a very unique signature which just kind of dies off, trails off like this. And you can fit that to a sine wave as well. But then, that gives you a signal where you have to know that's the signal of where it stopped. If you don't know that, then you kind of say, oh, well, that's an interesting mode of movement. And then you don't realize, oh, that's just where it stops. And so sometimes we have situations like that where we have to sort of take an image or take some data. We have to think about it as numbers or as like points on a graph and what those points on a graph represent. And sometimes it's interesting because sometimes if you work with data long enough, you get into like decades, especially certain types of data sets. You can actually see you see patterns of data points, and you know exactly what's going on. Oh, that's some behavior that happens. Sometimes you see it in like human behavioral data where you see some sudden rise in use of something. You say, oh yeah, that's because we talked about the bike share data set where we had, we saw that people sometimes don't ride bikes, sometimes they ride bikes at a peak time. And we kind of know from like the temperature and the time of day, oh yeah, they're riding bikes, or of course they would ride bikes at this time. So sometimes you get that sort of intuition. Yeah, this is just an example of that. So then we wanted to answer questions about movement generation, and this is just how it generates movement. It generates movement against a substrate, so it moves against the substrate, uses the cell wall to kind of facilitate the movement, and it's gliding across the substrate. And there are different proteins that it secretes as well that sort of lubricate the substrate so it can move. And so that's the sort of this standard hypothesis of movement. So, sometimes when we have a question or we have a hypothesis in machine learning or in data science, we'll often look to the real world system to ask, what do they think is the main mode of behavior? So, we might come up with a really weird idea about how this thing moves just by looking at the images or from our analysis, but it isn't really consistent with what people have done in that field, or said, this is basically how it works, or these are our candidate explanations. So sometimes if you go to the literature of your specific problem, you can find like answers to some of your questions about why the data looks this way. Okay, that's just a little bit more about the biochemistry there. And so in this project, we take microscopy data. We not only have a deep learning pipeline, but we have a physical modeling pipeline. Physical modeling, we can do dynamic systems type stuff, modeling physics. But in deep learning, we want to know the colony shape. We also want to know something about variation, position, and then something called topological data analysis, which is where you take shapes and I don't think we're going to talk about that this semester, but you can take you plot your data and they form shapes in the, say, bivariate plot, and you can characterize those shapes as higher order structure. So you kind of merge like data analysis with mathematical topology, and you're describing the shape of the data. That's a signal that you have. So differences in that shape of the point cloud or whatever are actually meaningful. Okay, so I'm going to shift to a different organism now. That was enough of diatoms. This is a nematode called C. elegans. So nematodes are very small worms. They live in soil, and that's their native habitat, and they usually feed off of biological waste, like rotten fruit or whatever. And they started using this model organism in the 1960s. For one thing, they were easy to capture and put into culture. For another thing, they had very short lifespan. And then they also they produced a lot of offspring. So it's pretty easy to do things like make mutants out of them, or to study their life cycle, or to study reproduction. And so what we're interested in is this developmental aspect, where you have a fertilized egg that divides into two cells. And then from there, it divides into a bunch of other cells. And those cells go through different movements, and they form this hatchling worm that hatches from an egg, and then goes through a bunch of larval stages to become an adult worm. So we can look at all of that pretty easily under a microscope by culturing these organisms. We have genetic data that we can maybe create mutants where there's a mutation in a certain gene, and sometimes that shows up in the phenotype. It shows up in the worm. Sometimes you have worms that are very slow moving. Sometimes they're missing like certain things in the phenotype in the body. It's a really interesting set of mutations we can work from. But in this case, we're interested in looking at these early stages of development. We want to track the cell nuclei, and we want to look at sort of the division of these cells and how they get up to about here, where they're forming a shape. So that's what a lot of the research is in these slides here. So interestingly, if you take an embryo, you can image an embryo like this. You see that you can see sort of the individual cells because they're clustered together in this clump, but they have contours between them. Those contours are the cell walls that are adjoining. And you see these little blotches, these pink blotches, those are the nuclei. So the DNA is the nuclear DNA. And we can actually use fluorescent proteins to light these up, to image these under ultraviolet light. So we can actually put things in the nucleus or get things into the nucleus, and you can put them under this kind of microscope, and it shows up as a color. So we can actually track these with machine vision. We can get these microscopy images and use what they call cell tracking software that just tracks these fluorescent signals, and that's used as your centroid of your feature. And then each cell is a feature. And then as the cells divide, you get more and more features. And of course, those cells that divide are dependent on the previous cell. So it's again this time series, time-dependent data. So we can build these models of not only from this two-cell stage, but even getting farther and farther along, we get more cells, and we can look at the division events and all these other things. So people have taken this sort of cell tracking data and taken advantage of a lot to build models of the So, if you have a couple pieces of information, you can build these models. If you have information about the nucleus of the cell or the centroid of the cell, of the feature, you have the cell contour data. So if we look at those contours in the original shapes and we turn it around, we can, sometimes we can get like what we do is we put our stacks of images. So from the top to the bottom of this, this I guess tube, or I guess it's like a beam, and then you just get images from different slices of that, and you can put them together so you can get shape and orientation data. So you can build these types of models where you have this three-dimensional model of the cells, and these cells don't look like cells under a microscope. They're just models of these features put together. And then, of course, we can model changes in those features over time. So, under a microscope, cell division looks like this. You start with a couple of nuclei, you rapidly get many nuclei, you get many cells. And as you can see, the cells are harder to detect as you go along. So the early cells are very easy to detect. If you go back to here, you see that the cell boundaries are easy to identify by eye. Here it's a lot harder to parse them out. So your machine learning algorithm is going to have to sort of deal with that. And it's hard to actually do something like a mass scare, because unless you know in advance what the cells look like. Oh, see, now it's turning into this comma. It's going through its comma phase where it kind of holds over. Yeah, so, and then as you get into this stage of embryogenesis, it's even harder to track the cells. So there are these problems like that. And a lot of times with data like this, it's just, it's a matter of really developing a technique to segment out all of the cell boundaries. And for that matter, all the cell nuclei. And again, the usual caveats about segmenting out the background apply here. In this case, we have this is an image. Okay, this is a fluorescent image where we have this supposed to be red and share, and it's in black and white. So you can just see it glowing versus not. So this is actually an easier way to extract information about these cells, extract information about where the nuclei are. So now we can actually take these points and segment out just those light points and say that's the center or a nucleus of a cell. And then we can actually use that to identify us. And then we can actually extrapolate a model here. We just assume that anything else, we'll basically use a DevoWorm, which we learned about, which is where we have these centroids, and then we find the sort of the midpoint between each centroid, and that's our boundary. And that's kind of how you can build these models. You can just use Voronoi tessellation, as they call it. That's a very technical term. It basically means you're applying the Voronoi process in 3 dimensions. You're saying every point, if you find the midpoint between those points, that's the boundary. And you do that for every 2 points until you get a bunch of boundaries that will fit. And so we're just basically taking a bunch of microscopy data that's maybe very hard to segment out, and we're finding a way to build a model. It's much easier to interpret and use for future analysis. So there are a lot of places you can get data for C. elegans. C. elegans is actually one of those model organisms where there's a lot of data. And one method that they have for C. elegans machine learning specifically is the cell tracking challenge. The cell tracking challenge is, or I guess it's still ongoing. It's a competition that research groups will join. And the idea is they have to track these cells. So like I said, there's software that allows you to track the nuclei, but there are all sorts of challenges to writing that software and to actually doing something with that cell tracking data. So this is the paper here where they talk about this, and there's a website, cell tracking challenge, where you can look at the the competition part of it, but also the data sets. So for these competitions, they'll get a bunch of data sets. Sometimes they get them from their own group, sometimes they get them from other places. And then they analyze these data sets using their own custom algorithms. and then they benchmark them on, C. elegans, and then you have a result, and the best model, the most predictive model wins. What we're trying to do is we're trying to predict a model of C. elegans of the cells from the cell tracking data. So that's one place to get data. There are other, this is kind of like cable, where we have data sets for all different purposes. And we have this competition around it, where people are just trying to improve their techniques and methods. And of course, in the process, they have a lot of data sets that they generate. This is the EPIC data set. This is more kind of a group of C. elegans researchers who generated this. This is it's kind of an older data set, but it basically gives you a bunch of positions for cell nuclei in the embryo with timestamps. So you can actually use this for, kind of as a baseline for your work if you want to model the embryo. So what we end up with here is we end up with this three-dimensional plot where we have this point cloud that looks like a bean. It looks like the type of thing we saw in the video and then we saw in the model there. And this is just a point model where we have the centroids of these cells, and we just show them in this space. And we have this volume, and then we're modeling this over time. So we have this time parameter, and then we have, if we look at like these data over time, each one of these points gives rise to two other points as daughter cells. So each point will divide eventually, and they keep dividing until at some point they stop when they reach the number of cells that need to be there. But these points will then divide and move to somewhere else in the space, and that's what we call the division. So this is a really interesting data set because you have this point cloud in three-dimensional space. But the point cloud is going to change and get denser as time goes on. And that's a direct result of these points ceasing to exist and dividing into two other points. It's kind of, it's not something you usually deal with in machine learning in general. You usually can assume that the cases are going to be static, but not in this case. And so we can actually build a model of this surface. This is a two-dimensional example of this where we might have one centroid that divides into two centroids, and then they keep dividing. They produce this map where you have 16 centroids, and you have information about all of these divisions. And of course, we want to be able to track all this in a machine learning context. So this actually provides metadata for us. These labels are cells that we know exist. We know the names of them. So we know when this divides, it's going to result in these two cells. We have a label for each cell. We have then the time that they're supposed to divide, and we know that they're certain orientation in space. So that's a lot of metadata to use for your machine learning analysis. Now with C. elegans, we have our developmental cells, we have every cell identified mapped. So we have the nomenclature name, we have the position. That's all known, and that doesn't vary across embryos. So it's actually, in this case, we use our model organism in a way that helps us here. It's very fortunate that we have these data, and it would say maybe C. elegans is a good model for machine learning because you have all the things you need for a successful machine learning. You have labeled data, you have a fair amount of data to work with, train your model, and you have the metadata that you need. And so we actually, in our C. elegans work, we actually we created 3 different models. We published them on Hugging Face, which of course, if you're familiar with Hugging Face, this is a site where you share code and share data for machine learning. So if you're interested in finding a data set or even a model, that's the place to go to find really cutting edge models. And we published a couple of ours. The first one is nucleus segmenter. So we're working on segmenting out nuclei from images. This is where you're just identifying the foreground from the background and showing this model. The membrane segmenter, which is a separate thing where you're not segmenting out these nuclear signals, but you're segmenting out the sort of the edges of the cells. So you have these membranes in this embryo. So that's another tool, and it's a different method. And then this lineage population model, which is where you want to look at these cells as they divide, and they divide again and again and again. And you could build a binary tree on this. So this is actually very similar. This is an artifact of biology, but this is actually very similar to our classification tree. And in fact, it is a classification tree because we start with a root, and we divide into two things, and eventually we end up with cells that are very specific in terms of what they do, and they have they all have names, labels, and then eventually this process stops at the end. So we could actually use a classification tree to understand this lineage tree, but we also need these other tools to augment them. And so this is the cell nucleus segmenter. Again, our microscopy images are going to be noisy by default. This is an example of something. So we have these fluorescent images where we have this light signal. And the light signal, you can see it's not clear in this image because there's a lot of noise that masks the light. So you have to identify what is the extent of this light signal. See, there's this thing in biology called autofluorescence where there's like a light signal that's a false positive. So you see some of that at the end. And it's just an artifact of different things in the biology. But basically what we want to do is we want to identify the extent of these blobs and then only capture these blobs in the presentation. That's what we're doing here. And sometimes you really need sensitive techniques that will say ignore the noise and capture a signal. Sometimes you need to use thresholds, light thresholds in the image. So with image processing, the image is basically just a color map or an intensity map of colors. And sometimes you'll have like pure black. Sometimes there will be a little bit of signal, maybe a different color with different colors involved. And then you just say, this is my cutoff for a signal. And so then you can just basically get a more sparse map with the things that you want. So that's the segmentation map. So there are other techniques you can use to segment images. Some people use recurrent neural networks. Some people can use watershed feature detection or feature selection. There are different algorithms that people use to segment images. Watershed is interesting because it's actually quite old. It comes from, I think, the late 60s and early 70s. And for some types of data, watershed works really well. It actually works on the principle of, like, if you look at a landscape, you notice that rivers have, like, water basically rolls down to the river basins, and they form these river basins. And the idea in watershed analysis, you take things that are kind of on the edge of each basin, like if these dots are basins, everything at the edge sort of gravitates towards them. So you're going to assume that like the most intense signal is the center of a blob, and then everything kind of going outward is part of that blob, and then you discount everything out to a certain distance. So you end up with these blobs that are sort of like these valleys, and then everything else is sort of not considered as a feature. So that's watershed. There are other techniques that we can use, but basically you have to play around with these techniques to get a good result. That's the bottom line. This is another thing that we're working on, which is a prompted segmentation model. So again, we have the segmentation model we can use. We can use labels for cells. We know that those cells have labels. We know where they're supposed to be. We kind of know their boundaries. So we can actually build a model, using different methods of segmentation. Well, we can also then, once we do that, we can use what they call this. Meta has a model called the segment anything, which is SAM. And that's actually a very, I think it's a very significant advance in feature selection because it allows you to segment things out of images very easily instead of kind of like this guessing. And so, it's an open source model, so you can fork it and develop it for different things. There's a fork of this called CellSam, which is used in biological research. So people often use that fork to do this type of analysis. This is where we use a bounding box, but also a label. So we actually attach a bounding box to this to find a feature, but then we attach a label, some sort of linguistic label. Now, why do you think we do that? Well, we want to be able to prompt it from, say, we want to be able to prompt it using maybe a large language model, or maybe some way to kind of describe it to the algorithm. So if you're using a different data set, you can describe it with linguistic features and it can go into another data set and identify those same conditions and pull out the data. So that's another kind of thing that's, kind of helps you. Sometimes you can build pipelines that involve different types of models. You don't just always have to use machine learning for everything. You can use other types of AI models. You can use other types of mathematical models as well. This is an embryo generator model. This is where we use a GAN, which is a generative adversarial network. This is where basically you have a generator and a discriminator, and they fight over, kind of what the image should look like. You end up with this GAN that generates different things from input data and then selects upon it. So these are embryos that are built using this GAN. So basically use nuclear segmentation data, put it into a GAN. And, about 700 years ago, GANs were a really big deal. And they've kind of fallen into maybe not so heavily, maybe later use, but they're still very useful for, generating things and generating images. And then, connecting that with other types of machine learning techniques. This is something we call the lineage population model. So we use some of this GAN work on this lineage population model. It's basically generating an embryo and then verifying it against real data to see how well it does. And it's actually doing pretty well. It's actually producing a smooth prediction that more or less matches real world prediction. Actually, I'm sorry, the prediction is noisier than the real world generation. So biology is generating the steps in cell divisions, and the model is predicting it like this, and it's kind of chasing the trend. So sometimes it's ahead of the trend, sometimes it's behind the trend, but it keeps up with the trend, which is what we'd like to see out of a predictive model. Like we saw with the regression models, where we had the prediction, we had the real world data. Sometimes it matches perfectly, sometimes it doesn't. And you have to sometimes play with the parameters of this model a bit to make sure it's making the proper prediction. And then this is where we're doing this for other sublineages and C. elegans, kind of getting a similar result. Okay, this is same thing. And so you can also use these types of, you can use machine learning in other types of pipelines. So this is where we use machine learning to segment cells, derive these point clouds, and then we might want to actually build graphs on this. So we want to know how these cells are related, not just in terms of divisions or which cells are next to one another, but what cells maybe are sharing signaling molecules or other types of things. So we can build these graph embeddings, which are actually really interesting. These exist in this higher dimensional space, so we're seeing the three-dimensional example, but they basically find different relational, give you different relational types of information that you can then use for modeling different processes in it. So you can actually do a lot of interesting stuff by starting off with doing machine learning on microscopy data, plugging into something like a graph embedding, and then doing predictions and other things at the other end. And of course, we can also do other things. We can apply this to building networks and building agent-based models, which I won't get into, but basically, we're just simply taking the data we got from our machine learning analysis training other models or building other models that are more informative to the biology that we're interested in or the biological questions. That's all. So that is what we've been doing, and I think that may give you, I think, a good indication of what you can do with machine learning, kinds of questions you might ask or answer, and then maybe some places to get data, or how people do once they get a data set.